Hello everyone. Today we will study pollen germination. Okay dear students, so first of all, the aim of our experiment is to study the pollen tube germination on slide. After that, requirements. What all we are required for doing this experiment? First of all, we need a compound microscope. Secondly, we will be required some freshly plucked seasonal flowers. We have taken China rose, Vinca rose and some other flowers also. So you must see here we required mature flower. That flower is not mature, so we'll discard it. So we required some mature flower. Pollen grains can be easily seen over there on that stamen anther part, you can see here. After that, what we needed? Here we require sucrose to make sucrose solution syrup. We have kept some sucrose in the small box. We need beaker to wash our brushes to make our sugar solution. We require the glass rod to stir while preparing the sugar solution. We need filter paper, blotting paper. We need glycerin, that is optional. We require a glass slide, cover slip, dropper, needle, brush is very much important. Now the procedure. The first step involves the preparation of nutrient solution. So for this, we'll take sucrose. We have kept the sucrose in the small box here. We'll take all this sucrose crystal, sugar crystal into this beaker. With the help of glass rod, we'll stir it well so that we can make a concentrated sugar syrup solution. After that, we'll take clean slide. One thing you should make sure here, the slide which you take, it should be cavity slide. In the middle of the slide, a cavity will be present here, so that when you will put the sugar solution onto the slide, it will remain there, it will stay there. So now we are taking the sugar syrup with the help of dropper, putting 2 to 3 drops of sugar syrup on the slide. The slide we should take for doing this experiment is cavity slide. Make sure that you use cavity slide for this experiment, pollen tube germination. Now, one by one, we'll take each flower. We have to dust the pollen grain of the mature flower from this stamen. You can see the stamen, the flower is mature. So on the surface of anther, you can see the pollen grain dust over there. So now what we'll do, with the help of a brush, we'll pat dry this brush little bit. After that, very gently we'll move our brush over the surface of anther. All the pollen grains will stick to this brush. After that, we'll dust all the pollen grains from the brush to that sugar solution. Like this, we'll do it again so that maximum pollen grains could be dusted over that slide. After this, we will wash our brush properly and we'll repeat this step again for dusting the pollen grains of another flower. So here we have from the middle, we'll tear this vinca flower like this. From the middle you can see all the pollen grains has been exposed here. So very gently we'll again use our brush and very gently we'll dust the pollen grains on the slide over that drop of the sugar solution. We'll tap our brush like this so that all the pollen grains which has sticked on this brush could be collected over that slide. Same thing we'll repeat for the rest of the flowers, but we should assure that a brush should be cleaned after doing this. Then only we can get the proper result for all the different type of pollen grains. Let the slide set for 5 minutes. Use of saffron stain and glycerin are optional. Here we are using glycerin because we have to keep our slides for 30 more minutes to observe the pollen tube germination. Like this, 1 or 2 drops of glycerin over the prepared slide. We'll put the cover slip over the prepared slide with the help of needle, making 45-degree angle with the slide. We will put the cover slip properly and will gently press it. We have to keep our slide very neat and clean, so excess of glycerin or the syrup solution will remove it with the help of blotting paper. Now our slides are ready to be observed under the compound microscope. One by one, each slide we can observe it under the microscope. So first of all, we'll able to see the pollen grains. Initially, we will observe numerous pollen grains characterized by presence of outer hard spiny exine layer and inner flexible entine layer. After 30 minutes, we will observe the pollen grains with pollen tubes. This is because of the absorption of sugary syrup by the pollen grains via the germ pore and emergence of pollen tube. This is germinating pollen grain. The pollen grains are also called microspores, produced by the process of microsporogenesis inside the anther. The pollen grains are characterized by the presence of outer exine layer and inner flexible entine layer. In between the exine, there present germ pore. On the stigma, when the pollen grain absorbs sugar solution secreted by stigma via their germ pore, there occur the emergence of pollen tube. This is called pollen germination. Here we come to the end of our experiment, pollen germination. This experiment was performed by my very sincere students Akanksha Misra, Swarnim Swati, Pooja Sharma, Pooja Kumari, Bharti Kumari and Ranjit Kumar of class 12th biology of Radha Govind Sri Secondary Public School, Ramgarh, Kant. My special thanks to Professor Amit Kumar. Thank you.